Let's see. Maria, can you take number 2? Not quite there. Go ahead and read it out loud and then just take a crack at it.
Why did you measure the transmitted for 5 minutes or on the sample?
Okay. 46.5.
What do you try to make sure of by measuring for 5 minutes on a sample that's got no enzyme?
You're going to unify the questions?
Yeah, it's the control. It's the control for the experiment. It's to make sure that it actually is the enzyme that's catalyzing the reaction, right? So that's our control. The second part of that question says, why did you measure the transmittance for each sample before adding enzyme? Fernanda, why? Why did you measure the transmittance before you added enzyme?
Because the transmitter went to transmittance.
Ben.
I don't know what you're talking about, sorry.
Why did you measure? Huh? For what? Pass? Pass. Why did you measure transmittance times 0 reading before adding enzyme?
Oh, is it going to get 0?
Because you're not that dang fast, right? You're not that dang fast. You can't get it in the machine that fast. That's why. Because it's not humanly possible to get a times 0 reading if it's not in the machine at the time that you add the enzyme. So we're not fast enough, so that's why we do it that way. Let's see. Scott, what's the active site on the enzyme? What does it do?
That's where it's able to connect and break apart the bindings.
Yeah, it connects. That's the binding. It's the part that binds the substrate. So that's the connecting. And it also breaks apart. It catalyzes the reaction. So the active site binds the substrate and catalyzes the reaction. Let's see. Stephanie, what is a substrate? Yeah, it's the reactant in an enzyme catalyzed reaction. Reactant in an enzyme catalyzed reaction. Gabriel, what suffix do you find on the end of most enzyme names? ASE, ACE, ASE. That is the suffix on the end of most enzymes. All right, question number 6. We already did this at the beginning of lab. What happens to the amount of substrate as the reaction proceeds? Decreases. What happens to the amount of enzyme?
Nothing.
Stays the same. Amount of product increases. Number 7. What does a catalyst do? Doug, what does a catalyst do?
Catalyst makes a reaction happen.
Yeah, so it facilitates or it speeds up a chemical reaction. Catalysts facilitate or speed up chemical reactions. Cross out number 8. Cross number 8 out, get rid of it. And metabolism. What is your metabolism? Vicky? Break down molecules. A lot of people think metabolism, they think breaking down. But metabolism is the sum total of all the chemical reactions in an organism. And it consists, we can break it up into 2 categories. We can break it up into anabolism and catabolism. This is all the reactions that break things down. This is all the reactions that build things up. Together, they are your metabolism.
You said sum total of all chemical reactions, right?
Yep, sum total of all chemical reactions in an organism is its metabolism. We can break it down into these 2 categories, anabolism and catabolism. And building up, tearing down.
Take it.
Right? And that's it. You already know how to do metric conversions by this point in the semester, I hope. So that one should be easy, right? Easy peasy. Lemon squeezy. So that's the questions. Yes?
Anabolism, is that the building up?
Anabolism is building up. All those chemical pathways that build big molecules. Catabolism tears them down into smaller molecules.